Good evening to all. My name is Deepika. I'm second postgraduate student, Department of Food Science and Nutrition, Graduate College, Mysuru. This study is based on the optimization of fermentation time and organoleptic evaluation of sorghum millet based batura. Basically, the fermentation is the biochemical process to obtain the ATP molecules with the release of oxidation of sugars and the release of acids and alcohols as an intermediate product. There are several types of fermented products in India, such as idli, dokla, appam, and other products, and the commercially based products, milk products like yogurt, cheese, etc. Sorghum and millets have been important staples in semi-arid regions such as Asia and African centuries. These crops are principal source of energy, protein, vitamins, and minerals, and other nutrients also. These can grow in harsh climatic conditions also. So it is called as coarse grain. It was also called as poor man's diet as it is cheap in cost and also nutritious in the terms of nutrients. And it is a gluten-free one than when compared to wheat, it is gluten-free and it is rich in fiber. Batura is a traditional fermented Punjabi dish which is served with different types of side dishes like lassi, chana masala, onion, pickles, etc. And the main thing is the sour curd is added to the dough preparation, which acts as a live inoculum in order to aid the fermentation. And this study was mainly based on the objective to develop the millet-based fermented product and evaluate its organoleptic properties to optimize its fermentation time also, and to develop a low gluten and glycemic food. The raw materials used in this study was procured from the local market of Mysuru. And these are the statistical tools which are used to analyze the statistical sensory scores and also the nutrient values. And the proximate analysis was conducted in the Department of Food Science and Nutrition in order to evaluate the nutrient content, how much it is present in the prepared product. And it was followed by the standard AOAC method. These are the product formulation. The main ingredients was the main base was maida flour, and it was replaced with the sorghum flour in different concentration, as you can see, in 100%, sorry, 0%, 20%, 40, 60, 80, and 100%. That is 100 gram of flour was proportionated in different concentrations, and other ingredients and parameters were kept constant. In addition to that, to aid the fermentation process, the baking powder was used with baking soda, and to leaven the product, the oil mixture, hot oil was used, and to smear also oil was used. The known amount of water is added to prepare the dough. This is the procedure to prepare the batura. Small wheat flour of different concentration with maida flour was taken, except curd and oil and water. All were mixed thoroughly, and then the hot oil was poured, and it was made into semi-hard dough. And the dough was allowed to rest for 2 hours. It was based on the research article. Standard procedure was chosen, and this dough was then taken and rolled into divided into small portions, and it was rolled into like a puri, and it was deep fried to until it becomes golden brown and crisp. And this were on the evaluation of its sensory qualities. We found that the appearance and the color, texture, taste, flavor, and overall acceptability of SB3, that is coded was sample of 40%, was highly acceptable in terms of sensory scores. And other rest were such as 60%, 80%, 100% was not so apt in acquiring such a highest scores in sensory aspects. When compared to standard, 20% was also highly acceptable, but as the objective was to incorporate millet in the formulation, so we chose the 40% has been chosen as a accepted one, most accepted one. And the effect of lactic acid fermentation on dough. So what is the use of fermenting a dough? That is normal puri and bhatura is quite different in aspects of fermentation itself. So what does it implies on that means the lactic acid bacteria, it can improve the macromolecule hydrolysis, thus enhances the digestibility and nutritional quality of bhatura, and also it increases the pH of the batter, and also gives the characteristic flavor for that batter. The substrate concentration, nutrient availability, and also how the flour and lactic culture is being used in proportionate manner is mainly dependent on the fermentation process. And the faster process, starch will swell and protein get hydrolyzed, and thus the dough capacity, swelling capacity increases. So the resultant batura will be is more appealing. The advantages of lactic acid fermentation. So as I explained that lactic acid fermentation is so useful in increasing the sensorial properties and also the nutrient availability. So what it does is the functional inoculum, it has emerged as a viable alternative for the chemically synthesized preservatives, as they can inhibit the food spoilage. It aids digestion process, improves texture of the food and flavor properties of the food, and it has also been considered as a novel wave of antagonistic against some of the foodborne pathogens like norovirus, calcivirus, and notavirus. The optimization of fermentation time. After the formulation of the millet with the Batura's standard procedure, the accepted variation was subjected to optimization of fermentation time. This was due to analyze that what the fermentation affects in the sensorial properties of that particular product. So as the Batura was formulated in different concentrations, 40% was chosen as accepted one. The optimization of fermentation time was varied by, as it was standard timings was 2 hours, it was varied from 1, 2, 3, and 4 hours respectively. And the same procedure and same formulations as of 40% was used to prepare the baturas, and it was subjected to sensory evaluation. And these are the sensory evaluation obtained for the optimization of fermentation time by variable hours, that is 1 hour, 2 hour, 3 hour, and 4 hour respectively. And the highest score was obtained for the third variation, that is fermented for 3 hours. It had a good appearance, color, texture, taste, and flavor, and also the overall acceptability when compared to all the variations. And this is due to the poor fermentation and intermediate fermentation levels. The 1 hour and 2 hours for Badura was not so acceptable. And due to over fermentation, the textural properties varied highly, so that the fourth variation was not so acceptable. The nutrition composition was analyzed by standard AOAC method following, and we found that the moisture was not so variable, and the carbohydrate level was variable when compared to the standard one. It reduced uh, significantly, and the protein level also increased significantly when compared to the standard batura. The fat did not get changed that much, and the fiber level increased significantly from the standard batura, and ash content also increased. Thus iron and phosphorus levels increased simultaneously. So these sensory scores and the nutritional value says that the fermentation and also the incorporation of millet can increase the nutritional level and also slightly decrease the carbohydrate level of that batura, and increase of carbohydrate and decrease of fiber, which it makes the food glycemic low. And also sorghum is a gluten-free one, so incorporation of a gluten-free millet can is the person's condition who is in celiac disease condition. And these are the batura which is of different variations. And this is the batura for optimized temperature, optimized fermentation time. So this is a summary that the standardization of fermentation time for selected variation, it resulted in acceptance of 3 hours variation, and 40% of the batura was highly acceptable in the formulated one with sorghum flour, and the poor fermentation resulted in the lowest sensory scores, while hyper fermentation also resulted in lower sensory scores in terms of all quality aspects in sensories. And it significantly increases the dietary fiber level and protein, iron, and phosphorus levels. Moreover, it also reduced the carbohydrate level slightly, and also it made that food as glycemic low. And the optimum fermentation time was found to be 3 hours. Thank you.